starting here. Okay, and then add to your notes, please. You're not doing anything else.
Get off Roblox, Diego.
Español, póngalo en la bolsa. La bolsa. What? That was bad?
No, miss,
At least I tried.
no, miss, but it was actually okay though. Like I understand.
Like you
what
understand
meant.
me, right?
Yeah, I understand what you meant. She said,
Okay.
oh, for your, like, homework.
You guys ready?
I was casing them.
Alright, so can somebody tell me how we build molecules? How do we build molecules? Dehydration reaction. You remove a hydrogen from one monomer, a OH from the other monomer. Those hydrogens and OHs, they combine to form water. Then you have a bond with the oxygen in between it, and then you form a dimer. If you do it again, you form oligomers, or three pieces. If you do it again, you form polymers. So you see right here, mono, di, oligo, poly. Those are just building up macromolecules. Opposite of building up, we have the breakdown of molecules, and those are called, that type of reaction is called, if we're removing water, that's dehydration reaction. If we're adding water, another word for water is hydro. Hydro. So you're adding water, that's hydro, and then lyse means to break. So if we want to break dimers apart, you add a hydrogen, a proton back on one side and a hydroxide back on the other molecule, and then it breaks. Okay, so know those two reactions. Dehydration reaction, opposite hydrolysis or hydrolysis reaction. Sometimes they call it dehydration synthesis or condensation reaction. All macromolecules do this, whether it's a carbohydrate, a lipid, a protein, or a nucleic acid. So on this one here, what is this? This is glucose, which represents an example of a monosaccharide. And with monosaccharides, we have the ability to make them disaccharides, like we just did with our model. Two glucoses stuck together is a maltose. So we built maltoses. But if we had glucose and galactose, which is another monomer, then we end up with lactose. And lactose can be hydrolyzed by lactase, right? So we could use enzymes to break and hydrolyze, but you guys also did a dehydration reaction to synthesize maltose. Guys, glucose, galactose, and fructose, they all look similar. So that's why when we built the glucose today, I wasn't sure if you actually made a glucose or a galactose because they look very similar, the isomers of each other. For sure, fructose looks a little bit different. How are they different? Fructose looks different than the other monosaccharides. This one is a 5-sided ring, and the other ones are hexose sugars, right? Here, you should know the examples of polysaccharides. So if it's a polysaccharide that stacks on top of each other, that is cellulose. Cellulose, like plant cell walls for its rigidity, for its structure, for its density. And then if it's branched or kind of helical, those are the foamier type of storage molecules. If you're a plant, that's starch. Fancy word is amylose. And then if you're animals, we store our polysaccharides as glycogen, and that one is stored in our liver. Okay, main purpose of carbohydrates is for what? Energy. And remember, how do we get energy off of these? Cellular respiration. You break off the hydrogens. So what's good about glucoses and sucroses and all these different polysaccharides is there's a bunch of hydrogens that you could get energy from. And that gets converted into ATP. Okay, our second category of macromolecules is, what are we looking at here? Proteins, which are made out of amino acid monomers. How did you know this was a protein? There's a Cu group on one side, or a carboxylic acid attached to an alpha carbon. On the other side, there's a amino group that has a nitrogen on it, right? And then on that alpha carbon, the last one that you should see is a little hydrogen. And then we said with amino acids, there's 20 different kinds of them. So that's represented by the R group. So I know there's not 20 different colors, but ideally there would be 20 different colors of amino acids kind of stuck together, represented by a ball. But here, this is kind of like a polypeptide chain. And this polypeptide chain is going to fold up to be a more complicated molecule, like a protein, that has a shape. So eventually all proteins have a certain shape. And if we look here, there's a link. That link gave you the ability to make macromolecules and explore their structure. So I threw that one back in. And with the amino acids, some of the R groups are positive, some are negative, some are polar, some are non-polar. So it gives them the ability to like fold and make a shape. If once, what do you think these are right here? These dotted lines in between, I see 2 amino acids interacting. That would probably be hydrogen bonds, right? And then other times you can form ionic bonds, which are a little bit stronger, to kind of make sure the shape is in the right order. Yes?
Because I know, because I clicked on the link, right? Why is there's like more than like, there's a lot more than I expected.
There's a lot more amino acids.
No, wait, never mind. This is a different link. This is the one I got.
Oh.
Yeah, we don't have that. We don't have that link.
This link I did from your old slides. We looked at chapter 2. It had this on chapter 2, so I threw it back on here because it's still related. That one that you have, I think it's from the book. So I threw in. Do you guys have another slide where it has? It looks like this. If you click the link, did it look like a polypeptide chain? So like Valeria, can I see?
Oh, the link?
Yeah. So if you have this link, it looks like a chain of polypeptides. I stole that one from your book, and I thought that one was it was under animations. So if you look on the electronic copy of your book, you have animations, and I thought that one was a good one to kind of show how proteins, once they're made into a chain of amino acids, that's not a functional protein. It has to fold up. So once it folds up and gets its right shape with its bonding, then it becomes functional. Okay, I need you guys to also know the name of this bond. So when monomers connect together in proteins, they're known as peptide bonds. So know that type of strong bond is a peptide bond. Peptide bond. Okay, the third macromolecule is a lipid. Peptide bond. The third macromolecule is a lipid. What's a characteristic of a lipid? Here I don't see nitrogens. We saw what? Carbon, hydrogens. Lots of carbons, lots of hydrogens, and that indicates that it is non-polar. So there are non-polar regions that make up lipids that give them this category. We know that these are called hydrocarbon chains in fatty acids. But can you spot the unsaturated fatty acid? this bottom one right here, there's double bonds because it's missing hydrogens. It's missing like 4 hydrogens, right? So this one is a unsaturated fatty acid. This top one is saturated fatty acid, and this will be a slightly different character than this one would be. This one cannot stack on top of each other, so this would be liquid. And these ones here, they can stack together and be more dense. And these are solid at room temperature. These are liquid, like olive oil at room temperature. These ones, they kind of clog up your arteries and veins, so they're not good to eat. Right here, I don't know if you guys can tell, I'm circling one fat molecule. So if these are monomers, I take 3 of them, and remember they have a Cu group? Do you see how the Cu group is right here? Like there's a Cu group, a Cu group, a Cu group. This is 1 fatty acid, 2 fatty acid, 3 fatty acid, and then it's connected to this 3-carbon backbone known as glycerol. So how did I dehydrate? Like how many times did it go through dehydration reaction to build a polymer? 3 times, right? So this hydrogen comes off, and this OH comes off. Yes? The OH comes off of the Cu group, and that hydrogen comes off of this alcohol group, and then it connected like that. Same with this one, and another one, and then it connected. Same with that last carbon. It had a hydrogen come off and an OH come off, and then it connected. And so this is known as a triglyceride, which is a fat. So you store fat this way. What's the purpose of these fats? What is the purpose of fats, guys? I'm pointing to hydrogens for a reason. It gives us energy. So you have like tons of hydrogen. If you imagine the glucose, it had C6H12, right? But these guys, they could exist like, imagine if you counted up the carbons and you multiplied it by 2, that's how many hydrogens there are. So if you could imagine the fatty acids, and especially the fat molecules, they store a lot of energy away. Fats are not the only types of lipids. There are other lipids. So on the side here, there are phospholipids. I think that one's a good one to know. Phospholipids, I think you guys have this on the next slide, yes? So phospholipid is very special because it makes up your cell membranes. So phospholipids make up our cell membranes, which make us all up. All organisms have these phospholipids that they arrange themselves in 2 layers. Do you see a layer on the top with blue heads, and then a layer at the bottom with also blue round molecules facing the bottom? Those blue areas represent the phosphorus head. Right there, there's a phosphate group. And this phosphate group is negatively charged. So if it's negatively charged and polar, is it gonna like water or not like water? This area is polar and there's charges. Water is polar. And it has slightly positive and negative charges. So this area likes water. That's why it's called hydrophilic. Hydro meaning water, philic is like the love of. So here, this area faces the watery part. of the cell. Oil is hydrophobic. So this area, do you see these two tails? What do you think these two tails are? They're hydrophobic, but what do you think these two tails are actually made out of?
Hydrocarbon chain.
They are hydrocarbon chains. They're fatty acids. So if you look at this, if you look at the molecular structure here, do you see the zigzag up, down, up, down, up, down? That's what you guys built on your fatty acid chain. They kind of just simplified it as up, down, but it's a carbon, 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 and then it's filled in with hydrogen, but they didn't even show it. There's two fatty acids here, and they call that the tails. So each phospholipid has a head and two tails, and the two tails are hydrophobic. So if you imagine a cell, if you have, I don't like the way that they drew this cell membrane, but it's actually a circle, right? If you imagine a circular cell, the inside of the cell is watery, and the outside of the cell is also watery. So how do we solve the problem with water on both the inside of the cell and water on the outside of the cell? We have phospholipid bilayer, so the parts that like water face the water. The parts that are awake, that don't like water, that are afraid of water, they kind of huddle to themselves in the membrane. Okay, we will have a focused chapter on just the membrane itself in chapter 3. And so in chapter 3, we're gonna revisit the cell membrane because we need to learn how things go in and out of it. Are you okay with phospholipids? It's another example of a lipid. It's non-polar, like a bunch of non-polar stuff right there. Yes, Valeria.
Miss, what does phospholipids make up?
Phospholipids, there's like hundreds of phospholipids here on top and on the bottom. It makes up the cell membrane. All of us are made out of cells. So the cells themselves are made out of membranes, and the membranes look like that. And if I were to go in more detail, they have a head and two tails. And the head is facing the water because it's charged and it's hydrophilic. The tails are fatty acids, and they're made of hydrocarbon chains, which make them hydrophobic because they're non-polar. Okay, I added this. So, can you guys add a slide? I realized that we left off one more example of a lipid. So the last example of a lipid is steroids.
Oh, cortisol.
Or, yes, so an example of steroid is cortisol. That's a stress hormone. Another example is cholesterol. We're all familiar with that one. We want to not eat cholesterol because it's very rigid, and then it's high chance. If you have high cholesterol, it clogs up your arteries and causes problems. But then there's also testosterone, which is very similar to estradiol. This is what guys have more of as a sex hormone. This is what girls have more of that makes us female. So females have more estrogen, guys have more testosterone. But notice there's an enzyme that just converts one to another. But these are all steroids. Yes,
What's the difference between testosterone and estradiol?
estradiol.
Estradiol.
Can you guys spot the difference?
Like the way it's formed?
It's deoxy.
So it has one less hydrogen and the oxygen is
The estradiol looks like it has one less oxygen. No, the oxygen's there, but it's an alcohol. I think this is what Jolly was saying. What'd you say, Jolly?
I said it was like deoxy.
I don't see it when you say deoxy.
Oh wait, no.
that means it lost some oxygen. So here I don't see loss of an oxygen. The oxygens are still the same, except one of them, testosterone, is double bonded oxygen, and then the estradiol, it's a OH group, so it's some sort of a alcohol. So because of that chemical structure, I think, and then there's like a methyl group. There's a methyl group on the testosterone. When I say methyl group, it's a CH3. So I see an extra methyl group on testosterone that I don't see on estrogen. But this is what guys have more and this is what girls have more. And those slight differences kind of signal other receptors to behave and grow different things.
Is it true that when the fetus is being made, it starts off females?
Yes.
And that
When we were all
males are-
made, we're technically, we all look like females.
So we don't know.
But then, so there's a little area, I wish I would have recorded, there's a little area that grows longer. And if you have testosterone, it's gonna bind to those receptors and grow the penis. But if you don't have testosterone, it's not gonna bind to those receptors and then it will not grow and it stays small as your clitoris for the females. So females will not grow the penis because it doesn't have as much testosterone signaling it to develop that area, whereas males would. And then we have also slight changes where the males will develop testes and females will have our ovaries, but they're similar in structure. Then they start forming other material inside their testes called seminiferous tubules, where they grow sperm. But in females, we have oviducts that kind of move eggs around. But it is a kind of like a chain of reaction where one hormone, and these are hormones now, one hormone kind of triggers a domino effect to cause other things to start developing. Okay, why do we call these lipids? I don't see fatty acid tails. Why do you think they call these guys lipids? Steroids are still considered lipids. I put in one more example, wax. Wax, you could tell it has hydrocarbon chains, right? And if you think about ear wax, when you wax up a board or you have wax on a tree and it's shiny, it prevents water from being escaped, or it acts as a barrier for water. So that one makes sense, right? That's non-polar. But why are these ones considered lipids? It's hydrophobic. It is non-polar. So the reason why they place these they're all looking like 4 rings, right? Those 4 rings of carbon, they kind of just move electrons around in the 4 rings equally, so it is non-polar.
So don't mix water with steroids?
Steroids and water, these are not soluble in water. So they travel into our bloodstream and then they float with the blood to its target cell. So some things are soluble, some are not. They will not mix with water. Okay, and that's the characteristic of a lipid. Okay, our last category, guys, I think we're on macromolecules now. Our last macromolecule is nucleotide. Our last macromolecule is nucleic acids, which are made of nucleotide monomers. Yes, Nicola.
Can you guys go over
The wax has 2 fatty acids, and it's non-polar. I don't have too much to say about wax. Okay, nucleic acid. So what makes a nucleic acid a nucleic acid? What atoms do you see? Nitrogen. Very good. Nitrogen. But the nitrogen, we call this area of the nucleotide a nitrogen base.
Oh, so it's like
So that section is a nitrogen base. Then there's two more sections.
phosphate.
There is a phosphate group. So here is, I think, one of the first macromolecules, apart from the phospholipid, where we actually see phosphorus being used. So phosphate group is phosphorus with 4 oxygens, like a cross. So if you see a phosphorus in the middle with 4 oxygens, that's a phosphate group. And we did see that in a phospholipid. But that's a second chunk of a nucleotide. The third chunk is a sugar. So if you see these 3 pieces, a nitrogen base and a phosphate group attached to a 5-sided sugar, that's a nucleotide. It should not.
it has to be a 5-sided sugar.
It's a 5-sided sugar. That 5-sided sugar, its name is ribose. Ribose sugar. You've heard of ribonucleic acid, right? That's RNA. You've also heard of deoxyribonucleic acid. What do you think that one is? DNA. Okay, so guys, DNA and RNA, those are both examples of nucleic acids. And I saved this one as my last macromolecule because I think these ones are, like, to me, the biggest ones. Because if you think about DNA, this is one base, one nitrogen base on a nucleotide. We have adenine.
This is like nitrogenous base always have to be adenine or
No, the nitrogen bases, there's
Adenine,
4.
thymine.
There's 4 kinds in DNA. A, G, C, and T. Adenine, guanine, thymine, cytosine. So A, G, C, and T. So there's like 4 kinds just for DNA. And these 4 bases, like, you could take one, this is a A, but imagine it connects to a T and another one, a G and another one, a C, A, G, C, T, A, G, C, A, G, take like billions, and then that's in one cell. So if you look at your DNA that makes up your genome, you take billions of these, connect them together, and but then you only have one strand. But DNA is double-stranded. So you take a bunch of them and you connect them together, and you have one string of nucleotides. Then you have a second string of nucleotides, and you kind of twirl them around in a helix, and then that's DNA. So like this molecule is DNA. Do you guys see red? What does that red represent? Oxygen, right? The black carbon. Can you see a ring? What do you think that 5-sided ring is? That's the ribose sugar, right? That golden ball, that's phosphorus. That's the phosphate group. So do you see how there's blue ones? The blue ones are, and adenine is one of the examples of a nitrogenous base, because it's nitrogen, right? And then it could be, if it's not adenine, it's guanine or cytosine or thymine. But if it's RNA, there's another one called uracil, the U one. But do you see how this is 2 rings?
Yeah.
I see a 2 ring like that. This one. This one is 2 rings. So like this piece right here, that's one nucleotide. And then this is another nucleotide. That's another one. That's another one. That's another. And then you have like billions. Okay, goodbye.
Okay, miss.